This is a presentation about the future of health, which will be in your stem cells. This is an outline of personalized stem cell banking and why this will become an important feature in therapy in the years ahead. So why bank your own cells? Why bank your adult cells and turn them into stem cells? There are many diseases like here we show an example of an adult motor neuron disease called ALS. And recently, Professor Stephen Hawking passed away from this disease. He suffered from this disease for decades, and there was no pharmaceutical intervention to cure him. This is one example of an incurable disease that stem cells could solve one day. The other is also a more common disease called Parkinson's disease. And again, a couple of famous people here, Muhammad Ali, a boxer, and Michael J. Fox, an actor. So Parkinson's disease is a disease where you lose your ability to control your limbs, and it's because you've lost some cells in your brain called the dopaminergic neurons. So we have ways now to create those dopaminergic neurons and inject them into animals to restore the ability to control movement. So this is cell therapy that's coming down the road. So what are the opportunities in personalized stem cell banking? Well, for example, there are many old people that will increase in the years to come. We call this the aging storm. And the developed world would have to change our way of thinking, says the former U.S. Secretary of State, Peter Peterson. And Innovate UK recently did a survey and found that although there are 15,000 centurions today, they suspect that in a few years from now, the 10 million people that are alive in the UK today are likely to celebrate their 100th birthday. So that's two populations of Singapore. A few years' time will be 100 years old and living in the UK. Stanford Center of Longevity shows that there are now 21 million people in the 60s, 65 to 75, and that will jump to 40 million in 2050. Those in the 75 to 85 range, 13 million will more than double to 29 million. And those above 85, right now are 6 million, will jump up 3 times to 19 million. Further, the global stem cell therapy market is set to grow to $170 billion by 2020. And there are 653, and this number is rapidly increasing, clinical trials using a form of stem cell called the MSCs, which is an adult stem cell. This is the statistics from the Alliance for Regenerative Medicine report at quarter 3 of 2017. You can see that there are about 40 phase 3 trials for gene-modified cell therapy and 45 in the cell therapy space. So if even 10% of them pass this phase 3 and become commercially successful, that's 8 new cell therapies that will come in 2018. So you can see this trajectory. In the past, the biologics field, which is the protein-based drugs to prevent and cure cancer, so different types of cancers, took about 20 years before there were 10 products. This is when our institute was set up in the 1990s. And now there are 60 new registered products a year up this trajectory curve. Meanwhile, it has taken us about 10 years, half the time, to go from one cell therapy product to 10 since 2000 and 2008, which is when our institute started looking into stem cell bioprocessing. So this curve is steeper and shorter in terms of delivering cell therapies to the population. So what is the brilliant stem cell banking offering? What is the value proposition? We are offering to the public for the first time the ability to turn your cells young again by converting them to embryo-like cells. So these are the cells that were made when your mom and dad came together and formed you in the embryo. So the offering is to reprogram your cells while you're still young and at the prime of your life, when you're disease-free, essentially, still relatively healthy. And then you store these pluripotent stem cells as a bioinsurance so that when you're older, they're available for you to heal a range of diseases. That's the value proposition of banking your cells now, as early as possible. So these are the pluripotent stem cells. You see here they're stained with different stains to show up proteins on the cell surface and inside the cells. Very colorful looking stem cells. So in 2012, a Nobel Prize was given in medicine for progress in stem cell research to Professor John Gordon from Cambridge University and a young Professor Shinya Yamanaka from Kyoto University, who's a peer of mine. So Professor Yamanaka discovered 4 genetic factors, let's call them transcription factors, that if delivered to any cell, almost any cell, can turn them back into these pluripotent stem cells. And for that, both of them were awarded the Nobel Prize. So where are the opportunities in actually using these cells? In the near term, 5 to 10 years, we will be able to have cell therapies to treat pain and disc problem, joint problems like cartilage repair, and blindness. So as you reach your 80s, you will lose your acuity of sight, and these cells will be able to cure that. In the midterm, in 10 to 15 years, you will be able to treat Parkinson's disease, which I described earlier, diabetes, and heart failure. These are much more challenging diseases. And then in 20 plus years, we will be able to treat complex diseases of the blood, dementia, and one day have rejuvenation therapy. And there's early indications that stem cells will be able to do that, both in mouse models of the disease, as well as individual case studies of patients. So is this science fiction or fact? Here's an example where a person with Alzheimer's disease, before he was transfused with umbilical cord blood, he had a lot of plaques, the white bits around the brain. And after transfusion, these plaques were reduced. So shows that it is potentially possible to even treat a complex disease like Alzheimer's with a stem cell transfusion. Here's an example from a mouse study where a certain type of cells, the neural stem cells found in the hypothalamus, when they were ablated or destroyed, it accelerated aging in the mice. So the reverse could be true, where if you could make these hypothalamic neural stem cells and inject it here, you could slow or reverse the aging process. So where are the future of therapies? As I mentioned, one of the earliest is using these retinal pigment epithelial cells for treating blindness, the adult mesenchymal stem cells for treating stroke, neurons for Parkinson's, neural stem cells for dementia, heart cells, cardiomyocytes, for treating heart disease, islet pancreatic cells for treating diabetes, and then for the highly complex aging process, blood and neural cells. So here's an example where a lady who was blind was infused with retinal pigment epithelial cells. These are the black-looking cells, and she regained her sight fully. So these cells are made from the pluripotent stem cells over time. Over a 40-day period, you get RPE cells from pluripotent stem cells, and then they're able to be injected into the eye. Very few cells, 10,000 cells, and restore the function. Company that's been around for 20 plus years, Athersys, is now in the late stage, phase 3 of stroke treatment. So they've completed a phase 2 trial, which was very promising, and are now doing a larger 300 patient phase 3 trial in Japan and the US. And I predict quite likely they will have the first blockbuster product for stroke using mesenchymal stem cells. Medipose, a Korean company, has already got a product called stem cell CAR-T-stem. So these are cells which are extracted from the umbilical cord blood and then injected into the cartilage to repair damaged cartilage. Brainstorm is an Israeli company that has also a mesenchymal stem cell, which has been engineered to make a protein called a brain-derived factor. And when these cells are injected, they help to restore the myelin sheath around neural cells and allow connectivity and communications, which is part of the problems in the multiple sclerosis disease. And they're in a phase 1/2 trial. So with these examples of therapies, there's also the example where cord blood from babies have been collected and banked to treat some of the blood diseases. So you can see the growth rates of banking have ranged from 9% in Singapore to 35% growth per year in Indonesia. And so the business model of banking is already workable. And globally, there are about close to 2 million units banked in the world. However, you cannot access cord blood for adults, so you can't bank cord blood as a storage option for adults. Also, in terms of reprogramming, the technology that was developed by Yamanaka is a lab scale process, and it cannot scale. So, brilliant research has tried to solve this problem by creating a technology that can expand the cells for therapy. So when you do a therapy, you have to go through small animal models where you need few cells, then large animal models like monkeys where you need 10 times more cells, and then finally the human system where you need even more cells. So the current methods of growing cells, stem cells, is limited to achieving materials for phase 1/phase 2 studies. And there's no technology to go to produce enough cells for the phase 3 and the market once they become commercially successful. You need billions of stem cells and then, for example, billions of neurons to be transplanted to thousands of patients. The lab scale culture is like this. You can see you take cells from patient, grow them up on a dish or a tea flask. They have limited surface area, inefficient, is expensive, and highly manual. So our solution is we will take blood, say 5 to 10 mils of blood from the donor, put on those 4 reprogramming factors and make human pluripotent stem cells. These human pluripotent stem cells we will demonstrate can differentiate to heart cells, neural cells, and possibly blood cells. We'll do this using a technology that was invented by my group in the Bioprocessing Technology Institute, A*STAR, and patented, called iPS spheres. So these iPS spheres will select a single cell, reprogrammed cell, onto the bead, and then they will proliferate over the surface of the bead. As they require more surface, they just attract more beads to there, and then they grow as a larger and larger cluster. So this technology platform we have demonstrated widely for a variety of applications. We have taken cells from different sources like T cells, skin cells, adult MSCs, blood stem cells, CD34 cells, and been able to reprogram them on this platform. We can also automate and close this platform so that it becomes an integrated process and amenable to good manufacturing practice. And this automation is key to offering the capabilities to the public. And once we've made these stem cells, the iPS cells, we can expand them up and then turn them to neural cells, heart cells, erythroblasts, which are precursor to red blood cells. We can also drive them to its adult mesenchymal stem cells, and these stem cells themselves can become cartilage, bone, and muscle. And these are also integrated into suspension and scalable bioreactors and can be closed as a good manufacturing process. So for the banking purposes, this is the process. We will take your blood or your skin, reprogram them, pick out the clones, which will be highlighted by a green stain, put a single bead onto the single wells, and then add more carriers, and then scale them up in larger and larger volume. So here's 5 examples of donors where we made 5 individual reprogram IPS lines, and we've also further made them into a red cell lineage. See the red pellets at the end. So the technology works by hand. We're going to turn this into an automated platform. So the whole process will consist of collecting blood and then testing for safety, and then the blood will be frozen down and stored. We will build this automation platform for reprogramming, and then expand up the cells over several weeks, test for the quality of the cells, and then harvest. And the last stage is freezing down the pellet of cells and storing them, which is a typical 40 to 50 day process. We will work with a consultant and automation companies to build it into a robotic platform so that it becomes hands-free from the manual process. So the automation will be held inside a closed system, a sterile environment, a single box, and this is the machine that we will be putting together. Then over a period of 2 to 2.5 years, we will create this GMP process, and we will be able to start banking and ramping up the numbers of customers that we can serve over the years. And somewhere in the middle here, we could franchise off this automation technology to other countries, other cities in the world. So an example of this has already been demonstrated where Sumitomo Pharma has announced the investment of $343 million to build a pluripotent stem cell plant. But you can see there are 4 people here. They have a manual process, 2 people doing it and 2 people checking it. So Brilliant Research will do all this for a fraction of the cost. So after growing up the stem cells, is an example of beating heart cells in the bioreactor, and we can take these heart cells and turn them into a heart sheet, and this sheet can be implanted onto the surface of a damaged heart so it can restore function of the heart. The banking will also work very closely with the Health Science Authority in Singapore. So here's an example of the building, both in Outram and in the BARPLUS. And they will regulate blood services, banking, and applied sciences. Recently, in April, I was in Tokyo at the Tokyo Center of Life Sciences giving a keynote talk about our automation platform. So the Japanese are also thinking about this, and in this building alone, there were the key societies such as the Smart Aging Center, Center for Aging Societies, Kyoto University, Osaka University, University of California, Japanese Pharmaceutical Association, Japanese Association of Medical Device. So they're seriously thinking about how the paradigm of cell therapy is going to change medical care. So finally, the team consists of myself, co-inventor of the technology, Professor Shaul Ruvani, visiting professor from Israel, founder and CEO of Gene Oasis, who will be developing the business, a future CEO, and from the medical side, scientific advisor, Dr. William Huang, who was formerly the medical director of the National Cord Blood Bank and now the medical director of the National Cancer Center. And 2 key staff, Dr. Alan Lam, who's the chief scientist who developed the technology, and Mr. Jin Lo, who helped me prepare some of the business aspects of this. We will also have key advisors from both industry and academia to drive the growth of the company. So thank you for your attention, and if you need to get any further information, there are 2 books that I've written, one on the stem cells basic education and one called The Authorities, which is a more general book about different aspects of lifestyle and living. Thank you.